Hello, everyone. My name is Cameron Myhrvold, and I'm an assistant professor of molecular biology here at Princeton. My lab develops CRISPR-based technologies for studying RNA, and today's talk is really going to focus on detecting viruses using CRISPR. Now, as it turns out, there's lots of different situations when we want to be able to detect viruses. And I've kind of highlighted these on the slide and color-coded them into different types of scenarios. And so in red, we have scenarios where we typically think about detecting viruses. That is to say, if you're a patient in a hospital or you go to the doctor's office or things like this. Now, during the COVID-19 pandemic, what we saw was an expansion of testing into the light blue areas, things like self-testing with those rapid tests or drive-through testing to increase the scalability as well as access to testing under those scenarios. But what I would argue is that we really need testing technologies that can scale up to the purple parts of this graph. That is to say, tests that return a result really quickly while also scaling to perform lots and lots of tests simultaneously. And this remains a major challenge. That is to say, we don't currently have testing technologies that can easily scale up to test an entire workplace or an airport or a school or a stadium full of people. But that's something that we're actively working on to try and address the problem. Now, as background, my lab works on CRISPR-based technologies for detecting viruses. And the CRISPR system is this really amazing bacterial immunity system that allows the bacteria to defend themselves against bacteriophage infection. And that starts with this CRISPR array that contains these repeat sequences interspersed with spacers. And those spacers ultimately get transcribed into a guide RNA that can recognize an invading DNA or RNA virus using complementary base pairing. And that's typically done with the help of an effector protein, which I've sort of shown as an oval. And one example of this that you may be quite familiar with is Cas9, which cuts double-stranded DNA that is complementary to a guide RNA. And so Cas9 is really amazing. But today's talk is actually not going to focus on Cas9. Today, we're going to talk about Cas13, which you can sort of think of as being the RNA targeting equivalent of Cas9. That is to say, like Cas9, Cas13 uses a guide RNA to direct it to specific target sequences, but those are target sequences in an RNA rather than a DNA. Also, unlike Cas9, Cas13 doesn't have a protospacer adjacent motif or PAM, and the spacer sequences are a bit longer to account for this. So they're typically on the order of 22 to 30 nucleotides long. And that's to give the enzyme additional specificity because it doesn't have a PAM. And then of particular importance to this talk, Cas13 has both cis and trans cleavage activity. That is, it can both cleave the target RNA, but also cleave other RNAs in solution. And we'll kind of get into how important that is on the subsequent slides and how we harness that to detect nucleic acids. But before I get into that, I just want to make the broader point that Cas13 has really similar potential to Cas9, but for RNA applications. And people have already used Cas13 to do all sorts of things, ranging from nucleic acid detection to RNA imaging to targeting specific RNAs for degradation or modification. And so this programmable ability of Cas13 to target RNA is really powerful. And that's something that my lab is really dedicated to harnessing. But like I said, today's talk is really going to focus on the nucleic acid detection piece that is so central to what we do. And so at this point, you may be wondering, okay, how do we actually use Cas13 to detect nucleic acids in the first place? And that's really illustrated on this slide, where you can see that Cas13, again, is binding to a target RNA with the help of this complementary CRISPR RNA. And once Cas13 binds to its target, it becomes activated. And then, as I mentioned before, it has this trans cleavage activity to cleave other RNAs in solution. And so what we're able to do is add a quenched fluorescent RNA reporter, which is shown on the slide, that initially does not produce any fluorescence. And this is basically a short piece of RNA that connects a fluorophore to a quencher. And so initially, the fluorophore is connected to the quencher quenches the fluorophore, you have no fluorescence. But when Cas13 binds to its target RNA and becomes activated, it can cut the reporter molecule, separating the fluorophore quencher, and producing a fluorescent signal that can easily be recognized with all sorts of different detectors that are out there. And so this gives you a really nice mode of signal amplification because it takes this one sequence-specific binding event and converts it into many, many subsequent cleavage events due to Cas13's catalytic activity. And this estimates vary, but this happens several times per second based on kinetic measurements that people have made. And so you can pretty easily see how over the course of a few minutes, now you've achieved hundreds to even thousands of fold amplification of the signal relative to conventional approaches, which really rely just on nucleic acid hybridization. Now, in addition to boosting sensitivity, as it turns out, these Cas13 or CRISPR-based diagnostic methods also boost specificity. And that's because the enzyme gets to decide when to become activated by the target. It's not just governed by the thermodynamics of base pairing. And so the enzyme can enforce essentially additional specificity based on its activation. All right, so that's the basics of how one can use Cas13 for nucleic acid detection. Now, in today's talk, I'm really going to focus on two topics. The first is really trying to simplify nucleic acid detection using this technology called SHINE that we developed during COVID-19. And then I'll talk a bit about some more recent work to use machine learning for CRISPR RNA design. And this turns out to be really powerful and really important because there's a lot we don't know about how these CRISPR systems work. They're a little more complicated than standard PCR primers. And so using these machine learning approaches, we can do a much more clever job of designing sequences that work really well for all of these challenging detection applications. And so we're going to start really by focusing on SHINE and talking about how to simplify detection and make it as easy as possible. And this really, like I said, came to the forefront during the COVID-19 pandemic. It's something we've been interested in for many years prior to that. And I'll mention very briefly at the beginning some of our early work and then kind of talk you through what happened during the pandemic. By way of setup, we need better viral diagnostics at the point of care. So what I'm showing you on this slide is a table that summarizes the existing standard technologies that we use for detecting viral infections. And these include qPCR, which is something that many in the audience are probably familiar with from the COVID-19 pandemic. On the other hand, you have things like rapid diagnostic tests or these antigen tests that are really easy to use. You can perform the test yourself and get a result in 15 to 30 minutes, but they're not as sensitive or specific as tests like qPCR. And so there's this fundamental trade-off, essentially, between the more complicated techniques that are laboratory-based and the simplified techniques that can be done anywhere. And our goal really is to get the best of both worlds. We think the ideal technology would have the performance of a nucleic acid test with the speed and ease of use of one of these rapid diagnostic tests. And so that's what we really set out to do when I started my postdoc, is to develop CRISPR-based approaches to detect nucleic acids in as simple a fashion as possible. And this really started back in 2017 with the development of Sherlock, which was led by collaborators of ours in Feng Zhang's lab. This is back when I was a postdoc at the Broad Institute. And so the way that Sherlock works is essentially it starts by harnessing the Cas13 detection reaction that I already explained to you previously, where we're using Cas13 sequence-specific recognition and cleaving reporters. And that sounds great, but on its own, it's not sensitive enough to detect the very low levels of nucleic acids that are present in clinically relevant samples. And so we need to add some things to the workflow in order to produce a diagnostic that has clinically relevant sensitivity. And those are basically summarized here on the slide. So we start with a sample that contains viral DNA or RNA. We then perform a nucleic acid extraction and do nucleic acid amplification, and then in vitro transcription to convert that double-stranded DNA into RNA that can be detected using Cas13. And we typically do that, I should say, by encoding a T7 promoter onto the forward primer for amplification, such that the amplified product can be transcribed in vitro and detected. All right, and this was sort of the state of the art as of when I started my postdoc, was this Sherlock approach. So we initially started by focusing on the extraction step and sort of saying, well, hang on a minute, this extraction step is a laboratory-based technique, and it's going to be really hard to do testing anywhere you want if you are reliant on extraction, because this requires, as I'll sort of show you on the next slide, a lot of equipment and laboratory infrastructure. And so we developed a method called Hudson to eliminate the need for nucleic acid extraction. And the way this works is basically it solves a major challenge, which is that all of our bodily fluids contain really high levels of nucleases. And these nucleases are very active. They're helpful in defending against invading pathogens, but they're very active and hard to destroy, and they will rapidly, within seconds, degrade any nucleic acid that's in your sample, making it really hard to detect pathogens directly from bodily fluids. And so the traditional approach, as I mentioned previously, is an extraction, where you use a lysis buffer, you bind a DNA to a column, you spin and wash multiple times using a centrifuge, and then are left with extracted nucleic acids. In contrast, the way that Hudson works is you simply add a mixture of chemicals, and in particular, there's a reducing agent called TSEP that is really at the heart of Hudson. And then you apply heat in two different steps, initially a low level of heat and then a high level of heat, and very rapidly, without requiring very much equipment, are able to process the sample in a way that is compatible with these downstream nucleic acid tests. All right, so we developed and initially published Hudson back in 2018 as a companion method to Sherlock. But during the COVID-19 pandemic, we realized we had to make it better. And that's because initially it was about a 25-minute method or so. And it had this two-step process, which is really important. The first step was really around inactivating nucleic acids while keeping the viral particles intact. And that's important because if you do things in the wrong order, then the nucleases will be there to chop up the viral genome that's present inside of the viral particles. And so this initial low heat step was inactivating all the nucleic acid in the sample, or sorry, all the nucleases in the sample. And then we would apply a high heat step to lyse the viral particles so that you could actually detect what was inside of them. But it turns out that for many respiratory sample types, if we add an RNase inhibitor to the sample, we can actually very much shorten that first low heat step. And this is really nice because it speeds up the overall workflow. And the performance, as I'm showing you on the bottom of this slide, is quite similar once you add this RNase inhibitor. There's very little RNase activity. On this graph, I should say, less fluorescence is a good thing. We're measuring the cleavage of an RNA reporter by nucleases that are present in different sample types. I'm showing you UTM, which is a swab sample matrix, as well as saliva. And you can see that if you do no treatment on these sample types, you get tons of RNase activity. Heat reduces it somewhat, but not to zero. If you only do Hudson, like the short version of Hudson, it's not enough. But if you add the RNase inhibitor, then you get down to very low levels of nuclease activity. We then had to greatly simplify the testing workflow. And so I explained to you how that works earlier in the talk. What we needed to do was basically combine several of the steps into one. And that includes the amplification as well as the in vitro transcription and the Cas13-based detection. And so that turned from a multi-step process into a single-step reaction. And you can see that when we initially did that, and I'm just showing you the data here on the slide, it really dramatically hurt the performance of the assay. That is to say, we lost almost 10,000-fold in sensitivity when we switched from a two-step procedure to a one-step procedure. And so then we got to work and we spent a bunch of time really intensively optimizing the biochemistry of the reaction. And I'm pleased to say that once we did that, we were able to recover most, but not all, of the gap between the two approaches. And we had to optimize things like pH, magnesium concentration, and primer concentration, and various other sort of biochemical parameters. And if you want to see more about that, you can look at our paper from 2020, where we described the initial version of SHINE, which basically, as I'm showing you on the next slide, is really a combination of Hudson and single-step Sherlock. And so this starts with either a swab or a saliva sample that you then inactivate using Hudson. and then perform this single-step Sherlock workflow that I just explained with either with a visual readout that is either a fluorescent in tube readout or a colorimetric readout on paper strips. And so all of that to say, we're really excited to develop this method. But of course, as the pandemic continued, we realized we had to make it even better than this, because while this is a big improvement over our initial Sherlock workflow, there's still quite a few steps involved here, and it's not as nearly as simple as a rapid antigen test. And so then later on during the pandemic, we developed this Shine version 2 workflow that is quite a bit simpler. So we came up with a rapid ambient temperature sample processing workflow, as well as figured out how to lyophilize, basically freeze dry all of the Shine reagents so that we could reconstitute them in the sample very quickly. And then you run the reaction and use a lateral flow strip for a visual readout. And so what I'm just going to show you on the next slide is how we were actually able to do that. So we came up with a new lysis procedure that involves a mixture of different chemicals that can basically inactivate the nucleases that are present in a sample at ambient temperature, as well as various detergents that can lyse the viral particles. And so with this new approach, we were able to achieve not a complete reduction in nuclease activity. I should say that's what's being shown on the slide here. It doesn't work quite as well as our original Hudson protocol, but it still works well enough for our purposes. We then developed this lyophilization procedure, and what I'm showing you here is basically that this allows us to have stability at room temperature for about a week or so, and at 4 degrees or minus 20 C for over a month, which really aids with the distribution and facilitates performing testing wherever you would like to perform it. Because one of the other limitations that I didn't really mention of Shine is that it is reliant, like many of these technologies, on a cold chain, and so we're trying to basically reduce the cold chain requirement through lyophilization. At the same time, it also greatly simplifies the reaction setup because you can lyophilize everything that you need for the reaction in a single tube and basically allow the user to simply add a certain amount of sample and buffer and resuspend that lyophilized pellet in order to set up the reaction. All right, so the other big thing that happened during COVID-19 that we had to really be aware of is that the virus itself started changing. And so in response to that, we developed Shine assays to detect different SARS-CoV-2 variants of concern. And the way that this works is basically the following. So we can design very specific CRISPR RNAs to detect different SARS-CoV-2 sequences. And if we have one CRISPR RNA designed for the ancestral variant and one for the derived variant that contains, let's say, a new mutation, we can then look at the relative activity of those two CRISPR RNAs to tell us whether or not a particular sample contains one strain of SARS-CoV-2 or another. And it turns out that this works quite well. As I'm showing you on the rest of the slide, you can see a set of samples that contain either Omicron or Delta. And on the left, you can see the Omicron positive samples all have greater activity with this derived CRISPR RNA, indicating the presence of a mutation that is specific to Delta. In contrast, the Omicron negative samples on the right-hand side of the slide all have more activity with the ancestral CRISPR RNA, indicating that they don't have that mutation. And this is something that's really hard, I should say, to do with other testing technologies because they don't have that combination of deployability and specificity to test for individual mutations at the point of care like this. One last thing that I'll just mention that we did in the context of developing Shine version 2 is also try and reduce the reliance on equipment. And we did this by seeing if we could get rid of the requirement for using equipment for heating. So I already mentioned we have an ambient temperature sample processing workflow, but we still need to incubate the reaction at 37 degrees in order for it to proceed. And so what we tested out in these experiments was actually incubating the reaction using body heat, basically under your armpit. And it turns out what I'm showing you is basically the sensitivity of the assay comparing incubation on a heat block, where we can control the temperature very precisely at 37 degrees, and then incubation at body heat, which is around 37 degrees, maybe a little bit less, depending on who you are and exactly kind of how warm you are. But all of that to say, we saw pretty similar performance when we compared the two approaches. Now, I'm not saying that anyone's going to necessarily want to do this. The reaction runs for 90 minutes, so you may not want to put it under your armpit for that long. But the idea was more to show that conceptually, we don't necessarily need equipment to provide heat for our reactions. All right, so that was kind of a whirlwind tour of some of the various things we've been doing with SARS-CoV-2 detection. I wanted to briefly mention that we've since been applying SHINE to detect all sorts of other viruses, including influenza, as well as other respiratory pathogens. And I'm really excited to continue using SHINE for all sorts of different detection applications. All right, now I want to switch gears a bit and talk a bit about the machine learning piece. And this is something that really came up by necessity because we don't know a whole lot about how the system works. And so our goals with this were really to come up with a way to predict how well a particular guide target pair will work. That is to say, how much does the sequence of a particular CRISPR RNA affect its activity? And then also, we know that many viruses have a lot of variability across their genome sequences. And so we also wanted to automate the design process to allow us to account for all of this variation in sequence that we know happens as viruses evolve and change over time. We also wanted to leverage simultaneously all of the sequencing data that has been generated. And there are now hundreds of thousands or even millions of viral sequences that have been published. And so we wanted to make it easier for people to come up with new designs, leveraging all of this data that is already out there. All right, and so to start, we gathered a giant data set using a different CRISPR-based approach that I won't really get into the details of, called CARMEN, which is a massively multiplexed approach for screening activity in a combinatorial fashion. And so we tested about 20,000 pairs of different guide RNA sequences and different target RNA sequences, and basically measured the activity of each of those pairs and asked, can we come up with a model that can predict activity based on sequence? And so we came up with a very simple machine learning model that initially classifies a guide target pair as either active or inactive. And then for the active ones, that is, that have an activity above a certain threshold, we do a simple regression to get an activity value that is predicted for that sequence. And then we compare it to experimentally measured activities and use that to try and learn general principles. And so how well does that work? We can do a pretty good job of classifying whether or not a sequence is active or inactive. And I'm just showing you here a plot that summarizes, how well we can do that. For example, if we look at the different quartiles of predicted activity shown here on the right side of the slide, what you can see is that if we predict that a sequence is going to have a very high activity, that is to say a distribution that is shifted to the right, we can actually do a pretty good job of that. Like our top quartile has the most active sequences, followed by the third quartile, number 3. What we start to see as the activity is lower is that there's a lot more variability, particularly in quartile 2. Some sequences are active, some are not active. And then in quartile 1, we have mostly low activity sequences. And this correlates reasonably well, but not perfectly, with true activity, with a correlation coefficient of about 0.68. And so we also validated, I should say, this approach using a completely independent data set that someone else generated using Cas13 to knock down RNAs in cells. And it turned out, again, there was actually a pretty good correlation between our predicted activity using a completely different set of measurements and the RNA knockdown level that was observed. And I should say there's a negative correlation here because they were measuring RNA knockdown, we were measuring activity that turns on in response to cleavage. So we were expecting a negative correlation between the two, and indeed that's what we saw, was a negative correlation with a pretty high correlation coefficient. All right, so the other big part of this problem is that many viruses have highly variable sequences, and I'm just showing you an example here for Lassa virus, where you can see there's tons of mutations throughout sequences that were collected over time. And you can see this for other viruses too, like enterovirus B also has a ton of sequence variability. And it's really hard to come up with sequence-specific designs to target these viruses because they vary so much in sequence. And indeed, if you try to do that, you often only detect a very small fraction of the sequences. But if you use our machine learning approach, and that's shown in the colors relative to the sort of browner colors, you can see that our optimization approach covers a much higher fraction of the sequences than these baseline approaches that are based on a consensus sequence or the most common sequence that is present. We also automated this for designing assays against 1,933 different viral species that infect vertebrates. And so we were able to scale up our approach massively. And most of these designs progressed very quickly. 80% of the designs took less than 2 hours to complete. Within a day, we had designed all but 3, and the longest took about 38 hours from start to finish. So we think that's pretty remarkable that this approach can be scaled to such a degree. We also did some experimental validation using some of these highly diverse viruses, and I'm going to show you just one example here, which is enterovirus B. And here what I'm showing you is a bunch of different targets that represent the viral diversity of enterovirus B. And what you can see is that the ADAPT approach performs much better than standard approaches in covering across all of that diversity. So there's basically more purple across the sequence diversity than with these other approaches. And this is particularly evident at lower input concentrations, where you can see that the ADAPT designed CRISPR RNAs, which are shown in green, do much better than ones designed using a baseline entropy-based approach, which is just based on sequence conservation. So we have made all of our ADAPT designs available as a resource online at adapt.run, where you can browse all of these existing designs that we've come up with for all sorts of different families of viruses. You can also look at where those designs are in the viral genome. We made a graphical user interface and a browser, so you can see where the sequences are located on the viral genome. And we hope that this tool will be useful to people who are trying to come up with their own designs. And I should say, if you want to come up with your own as well, we also have a cloud-backed interface for running ADAPT, where you can upload data either from the NCBI database or upload your own data, and then set various parameters and do the run all in the cloud. And so again, you can also, I should say, we've also made the code available on GitHub if you want to download it and run it yourself on your own machine. But if you don't want to download it, you don't have to. You can run it through the cloud. All right. Now I just want to take one last moment and say that we didn't stop with ADAPT. We've actually spent some time more recently working to use generative algorithms to improve the CRISPR RNA design further. And what I'm showing you here is basically just a teaser of the results of doing that. And so we considered a couple of different approaches for doing this that are shown in blue and teal. And basically what these approaches do is generate new CRISPR RNA sequences that are not observed in nature. And that's not something we had really tried before. Previously, we had basically asked, okay, if you look along a viral genome sequence, where are the spots where I'm going to get the most active CRISPR RNAs? But again, almost always using sequences that were designed to pair perfectly to the target. And this makes sense most of the time when you're doing design, but sometimes actually adding mutations into the CRISPR RNA can make them either more sensitive or more specific. And that's what we've been able to do with some of these newer generative approaches. And I would encourage you to check out our recent paper in Nature Biotechnology describing some of these generative algorithms and how they can do a much better job with nucleic acid detection. All right. I just wanted to close by acknowledging a number of people who did the work. This work was led by a number of graduate students that I worked very closely with during my postdoc. So Catherine Frigi and John Aristi-Sans really led a lot of the experimental work, developing the SHINE technology. And then Hayden is really the brainchild behind ADAPT, and Srikar, who is another mentee, worked very closely with Hayden to develop these more recent generative algorithms that I just talked about. Thank you so much.